Design of an expression vector to create a human prostate cancer cell model. What is the objective? The objective is to create an expression factor to express PC4 into epithelial prostatic tissue to study its function in prostate cancer etiology. Why? The justification is that PC4 has been found to be an important factor in the development of castration-resistant prostate cancer. Also, in the development of metastasis in the same disease. The positive cofactor 4 or SUB1 cDNA sequence is shown here. The vector design was done using the plasmid called PMAX-GFP, which has a CMV promoter, an EGFP reporter, and a neomycin resistance for eukaryotes and kanamycin resistance for bacteria. The forward and reverse primers have a flanking DNA or overhang for the restriction enzyme called SAC1 or SAC1. There is also no ribosomal binding sequence because the plasmid already has the Kozak sequence. In order to obtain the desired vector, we have to perform 3 steps. The first one is the PCR of the SUB1 DNA with the same primers, which have a restriction site for SAC1. The second one is the digestion of that PCR result with the PMAX-GFP using the restriction enzyme SAC1. And the third one is the ligation of both. As the plasmid has only one restriction site for SAC1, there can only be 2 results. A plasmid without the inserted sequence, and the second one is the desired one, is the plasmid with the inserted sequence in the same direction as the promoter. As you can see in the image, that will be the structure of the sequence or the plasmid that we want to obtain, where we have the GFP first, and after that we have the SUB1 gene inserted in the same direction as the CMV promoter. To perform an E. coli transformation, a transduction of E. coli competent cells, we have to do another 3 steps. The first one is to incubate the ligation process, the resultant of our process from our previous steps. The second one is to perform a heat shock of E. coli cells for transformation. This is done maintaining them on ice and then at 42 grades Celsius for 30 seconds. And the third one is to culture them in agar plates with kanamycin. To perform an E. coli transformation, a transduction of E. coli competent cells, we have to do another 3 steps. The first one is to incubate the ligation process, the resultant of our process from our previous steps. The second one is to perform a heat shock of E. coli cells for transformation. This is done maintaining them on ice and then at 42 grades Celsius for 30 seconds. And the third one is to culture them in agar plates with kanamycin. To transfect the human prostate epithelial cells, first we have to extract the plasmid from the Escherichia coli transform because these ones have the desired plasmid. Secondly, we should do a vector transfection. This vector transfection is done by firstly using the result of this extraction of plasmid. First you put minimal essential medium and you add a transfection reaction such as Fugin or something like this transfection reagent because there's a lot in the market. And in order to know if the gene is being expressed in our cells, we have to look at the GFP, at the fluorescence of the cells. This could be done only by eye or with other techniques.